All right, so this week we're moving on to topic 5.4, which is cladistics. So in topic 5.3, we started talking about taxonomy and how there are all sorts of different kinds of life and that we needed some way to classify them. Remember that we have different taxa, these are different levels of organization that run from the very broad of domain all the way down to the specific of species. Let's always remember our little mnemonic. Did King Philip come over from Great Spain? To remember the order between those. Remember that all living things have at least one or, I can't spell, one or more cells. That all living things have some sort of DNA, whether that's in the nucleoid of a prokaryote or in the nucleus of a eukaryote, that they all have those A's, G's, T's, and C's, that every living thing uses mRNA and protein with the same 20 amino acids. All living things use ribosomes to make those amino acids, and that all living things have cell membranes with at least phospholipids in them. So that is true of every domain. Remember that prokaryotes are always single-celled. They have that cell wall. That is true of some of the eukaryotes as well. But prokaryotes will both have the single circular chromosome, and they have every way of getting nutrition and every way of reproduction. We have the different plants, non-vascular, which is only the bryophyta. Think of that as like broccoli. So these are the ones that don't have any stems or roots really. They reproduce by spores, and because they were the first land plants, they do have to stay in or near water because they cannot transport water throughout their body. So this is in contrast to the vascular plants. So these are the plants that have the stems and the roots of the leaves. This is probably what you're more used to looking at. So they come in three flavors: the Phyllocinophyta, which are like the ferns. So just think F fern. They reproduce by spores, and you will only find them in watery environments because their gametes do need water to travel. After that, we have the Coniferophyta, which has the seeds in the cones and those needle-like leaves. And then the last vascular class, the largest one, is the Angiosperms, which has the seeded flowers. We talked about many different animal phyla. So the standouts are Porifera, which is the simplest kind. It does not have symmetry of any kind nor segmentation. It doesn't have really a digestive tract of any kind. It is a filter feeder. Cnidaria is the only one with the radial symmetry, and the other giveaway is that it has those stinging cells. And then the Platyhelminthes and the Annelidas, these are your worms. They both have bilateral symmetry. The difference between the flatworms and like the earthworms is that the Annelida is highly segmented, whereas the flatworm is not. And the flatworm has a mouth but no anus, but the earthworm, the Annelida, does. So keep track of all of those recognition features. So now we're moving on to cladistics. So cladistics is a system of classifying organisms based on traits that are derived from common ancestors. So for cladistics, we might be staying in the same, family or order, but most likely we're going to be skipping around from different taxa and dividing them up based on features that they do or do not have. So this is showing kind of ancestry and traits at the same time from a number of different organisms. So you might see a cladogram in one of two forms, either this tree-based one on the right or more of like sort of a boxy one on the left. And so what is dividing them are these different traits, like say bony skeleton or eggs with shells, or on this side we've got traits like walking legs or hinged jaws. And what a cladogram is depicting overall is some sort of evolutionary history between these different organisms, and it's showing more or less when they diverged away from each other. So not like an actual year, just compared to one another, how far ago or how far long ago did these animals diverge from one another. So how cladograms are constructed is based on the presence or absence of something called a derived trait. So remember that we believe that all life originated from a single-celled ancestor, something very simple like a prokaryote, and over time living things evolved into sort of these higher order forms with characteristics, with traits. So these traits did not exist originally, they were quote unquote derived. And so how cladograms are broken up is based on these so-called derived traits. So somewhere along the line, something was, it evolved to have hair, and the organisms that are downstream of that ancestor have hair. So in this case, the primates and the rodents, they both have a derived trait of hair. So that's like one derived trait that is on this branch. On this branch, we have a derived trait of eggs with shells. So everyone that is downstream of that branch, the crocodiles and the birds, they have eggs with shells. But if we go further back in evolutionary history, right, someone had to have had an amniotic egg. So this derived trait is shared by all the animals that are downstream of this. So the primates, the rodents, and the rabbits, the crocodiles, and the birds, they all have amniotic eggs. So same thing with the forelimbs. The amphibian plus all those other animals, this trait was derived. It did not exist when you were talking about a bacteria. There was some ancestor back here that was the first to have forelimbs, and then all of their descendants got it. So that's what we mean by a derived trait, right? So derived traits come from some sort of shared ancestor. So we have, birds here. There was one organism that was the first to have feathers, that was the first to have beaks like this. That's different than a shared trait, right? So all the birds have wings that are similar to one another. They derived that from a shared ancestor. But a shared trait, a waspbill has a wing, a bat has a wing, and this hawk kestrel thing, it has wings. But they are different structurally. We can assume that they got them from different ancestors. So that's not a shared. So shared trait is different than derived. Derived from a shared ancestor. Shared trait means it evolved separately from different ancestors, okay? So derived trait is what we're tracking on cladograms, not a shared trait that came from a different ancestor. So let's talk about some of the structures that you see on these diagrams. So we have different organisms, again, that are split up depending on whether or not they have a certain derived trait, and as you move on, the list gets smaller and smaller. Each branching point, so these are called nodes. So each branching point, these nodes, is representing some shared ancestor where a species diverged. So somewhere along the line, there was some ancestor that was the ancestor to all of the organisms that are in the cladogram. But over time, again, the species move away from each other. So we have a shared ancestor for everybody, but then we have one ancestor that develops a vertebral column. And that ancestor would be right here in this node. And so this ancestor is basically like, I don't know, the grandpa to everybody else on this side. And so everyone that is downstream of that has that derived trait. That shared ancestor is the one that first evolved the vertebral column. As we go along, a different ancestor, separate from the one that went this way, evolved a hinged jaw. So therefore all of its descendants have a hinged jaw. So each of these nodes represents a separate ancestor that has a separate derived trait. So those are the nodes. So as we talked about in genetics, DNA mutations are happening all the time, which lead us to protein sequence changes. And so each of these nodes, yes, it's an evolutionary change, but remember all of those changes are rooted in DNA sequences that give rise to differences in protein sequences. And it's giving you an idea of when that divergence occurs. So this is not like in exact years or anything. It's just that we know that this divergence happened first. So the lancelet diverged from the others first, and then as you go down the cladogram, those represent more recent divergences. So the turtle and the leopard, because they both have amnions, they diverged from one another later. This is the last divergence to occur. So again, nodes, common ancestor for everybody that's downstream, everybody that's a descendant. They're going to represent some sort of change, some sort of mutation, right? Because they got those new features. And it's a relative estimate of the time of divergence. Oldest ones are at the top, youngest ones are going to be at the bottom. So if we look at this cladogram, so we have a bunch of different insects, right? Or I guess kinds of ants. And we are looking at the geez, Dorlemorphus, and the Formicinae. So who is the most common recent ancestor between these two? So how you do this kind of problem is like literally put your hands on the two starting animals. So like actually, take your fingers and line them up on this red insect here and the Formicinae. And what you're going to do is you're going to go backwards. So you follow the line backwards. So for the Formicinae, if I have my hand here, I'm going to go backwards and I'm going to stop at a node. And then I'm going to separately put a finger up here and I'm going to go back and then stop at a node. So then I say, okay, am I at a shared common ancestor? So for B, that is a shared ancestor. I can trace it down to both of them. So I'm going to say that the shared common ancestor is B, and it is. So it's not C because this is referring to this node. So if I went back here, C is the ancestor to these three here. So this is not a shared ancestor for either one of them. So it's not C. And D, if I just went back one, this is an ancestor only to this clade right here. So you can just sort of trace them. So the only shared ancestor for both of these is if I go, it's not here, I go back one, still not there, and if I go back one more, then I can trace it directly to both. So the answer for that one was B. And A and E are so far off in the distance, you wouldn't even worry about that one. The other version of a question that you might see is, who is the most closely related to a certain species? So we're looking for Myrcinae, which is here. So who's most closely related to this guy besides this one? And so you basically count nodes. And remember, the nodes are shared ancestors. So if I have fewer shared ancestors between myself and someone else, that means that we are more closely related. So the I can't say these terrible words, the Myrcinae and the whatever. Between these two, they're shared ancestors right here. So they're closely related. But the next one is going to be this guy right here, the Dolichordinae. So this would be the most closely related besides the one right next to it. And it's just because it is 2 nodes backwards. So that's going to be the most closely related one. So let's just look at another example right here. Who is most closely related to the elephant shrew? The wombat. Let's see, where's my wombat? So the wombat is back here. Or the armadillo, which is over here. So the thing to be careful about is to make sure that you're not always just choosing the one that's physically closest to you on the list. Remember, we have to count nodes. So if we're going to go for elephant shrew, let's trace our way back. And so let's say we're going to count the nodes between elephant shrew and the wombat kangaroo. So I'm going to go, okay, 1, because I just traced this line to here, so that's 1 node. We're going to come back 2 nodes. I want to go up that way because that's where the kangaroo, the wombat is. So I said, okay, 1, 2 nodes, trace, come back 3, come up, this is 4 nodes, and I come back and that's 5 nodes. So it's 5 nodes to the wombat kangaroo. So how about to the armadillo? So we had 5 for wombat. So I go elephant shrew 1, come down 2, come down 3, and that got me to the armadillo. So the armadillo is going to be more closely related because there were fewer shared ancestors between them. So even though it looked like the wombat was right next to that elephant shrew in the cladogram, it was more nodes that it took to go backwards and to actually connect that, right? So elephant shrew was here, 1, 2, 3, 4, 5. There were 5 nodes separating the wombat, and then it was only 3 for the armadillo. The armadillo is more closely related. So, so that's the nodes and how you would use them to to find the most a recent shared ancestor or to tell how closely related or not something is. The other feature of a cladogram that you do have to keep in mind is the root. So the root, again, all nodes are representing some sort of shared ancestor back in evolutionary history. It's never, labeled, but we're just assuming that it's there. So the root is what's down here. So this is the shared common ancestor for all members of the cladogram. And in this case, the shared ancestor has the derived trait of vertebrae. So everyone that's downstream of that is going to be a vertebrate. And again, the first to diverge. So this is not in number of years. The first to diverge is going to be closer to the root, and the younger, more recently evolved are going to be further down. So that's not to say that birds are a younger species. It just means that on this cladogram, they diverged more recently from these other animals than the ones on this side. So the shark diverged earliest compared to these other animals, and the birds and the crocodiles diverged from each other most recently. So older divergences on this side, younger divergences on that side. Root is the ancestor of them all. So we refer to these groups of animals as clades. So a clade is any group of organisms that share a common ancestor. So to share a common ancestor, you have to be connected to the same node. And so when they are like this, we call this a clade. So this is a clade because they share that common ancestor. This is a clade. It's just the same picture that's highlighted larger, but they also all share a common ancestor. But these two, this is not considered a clade because we're not including the nodes here. So the clade is the common ancestor plus all of the descendants. So yeah, we got some descendants, and yes, there is a shared common ancestor here, but not all of the descendants were included, so therefore it is not a clade. So clade is both the shared ancestor and all of the descendants. So cladograms, again, are created by dividing these organisms into whether or not they have those inheritance traits. And how we make them is sometimes you'll start with a table here, and they'll list the organisms, they'll list certain derived traits, and then they'll mark whether or not they possess those traits or not. So you will sometimes be asked to construct a cladogram from a table just like this. So you start with the organism that basically only has one derived trait. So one trait in common with everyone else. It's going to be the most different. So that is the organism that you put at the beginning. So if we look at this list of organisms, and they were nice that they put them in order for you, and they have these different derived traits. So like whether or not it has jaws, whether it has lungs, hair, no tail, bipedal. And for each of these derived traits, each organism will get a score, like does it have it, does it not have it. And as we can see, for the lamprey, it has absolutely none of these traits. And then the shark has only one. It only has jaws, but sharks don't have lungs, they don't have hair, they're no tail. That seems not correct, but whatever. A shark is definitely not bipedal. So how we'll organize this is we'll start with the one that has the fewest derived traits, which is the lamprey, how he ends up here. Next, they're going to put the shark. I don't know that I agree that a shark doesn't have a tail, but whatever. So shark has only one derived trait of jaws. Next up is the salamander, who has both the jaw and the lungs. So you'll just order them like that. So this is the picture of how it goes. But now let's try to organize some cladograms. So you can do this with DNA sequences. So I have a list of DNA sequences here. You start with the one that is the most different, that has the fewest shared things, which to me looks like number 3. So I'm going to guess that number 3 is the first one. And then, okay, this one has 3 differences. Now let's go to 2, So it would be 3, 2, probably 4, and then 1, or I have that completely backwards. I'm wondering, we'll have to look at that one. All right. So when you're constructing a cladogram, you might have different organisms, and you have to sort of look at things about how they're the same or they're different when, to look for traits to separate them. So I've got a bunch of different organisms here. To me, this little caterpillar here looks the most different. He doesn't have appendages of any kind, right? So, oh, I have their names here. So let's start out by looking, what is shared, what is different. Some of them have eyes. I think all of them have eyes, right? So they all have eyes. So then the next discriminator is, do they have feet? The cocoon is the one that does not have any feet. We can also divide them on whether or not they have a mouth. The cocoon does not have a mouth. Neither does this omanite. I don't know how you say this. But he does have some feelers, which is, feet-like. So for sure, we're going to start with the cocoon. We'll probably put this omanite second, and then bellsprout and Balbazar, right? So that is how you would start to separate them and kind of like make your little cladogram from there. So when cladistics first started, again, people were big into taxonomy in the 1800s. They were going only by physical traits but that you could actually see. Since then, we've had these advances in DNA sequencing and protein sequencing, and this has led to the reclassification of some organisms. So old school, right? Old way of classifying was based only on homologous structures. So things like the pendactyl limb, where you have those different purposes, but like, swimming if you're a whale, or flying if you're a bird, or grasping things if you're a human. So those are different purposes, but the structure is similar. We also look at like vestigial structures, so things like tailbones. This is how we used to classify things. But now we've got advanced technologies and we can use DNA sequences and protein sequences to do this instead. So one of the classifications that they got really wrong was figworts. So this used to be thought of as one of the largest families of angiosperms. So they're, vascular plants, they're angiosperms, they have these sort of small flowers. And it wasn't until they did DNA and protein sequencing that they realized that actually there's 5 clades that were originally classified in this family, and they had to separate them out. So it was only because they were looking at those DNA sequences that they realized they weren't as related as they thought. They had to combine 2 different families. 50 genera were moved to the plantain family, so those are like the little banana looking things. So those reorganizations are probably going to become more common over time as they sequence more and more organisms. So just a reminder that DNA changes are occurring all the time through random mutation, and so we can use those DNA sequences to reorder them. So that is it for cladistics and the end of topic 5.